Hello, my name is Ivana Fontes. Today we'll be discussing one of the many experiments that was used to find the structure of DNA, specifically Chargaff's rules. Chargaff's rules. What are Chargaff's rules? Number 1, the percent of adenine is approximately the percent of thymine. The percent of guanine is approximately the percent of cytosine. DNA composition varies species to species. For example, humans have more adenine thymine than turtles, and this varies for all the species. How do we know this? That is thanks to these two. Mary Daly and Erwin Chargaff. Daly developed methods for the extraction of nucleic acids and the relative amounts of purines to pyrimidines. Erwin used Daly's methods in his own experiments and found out Chargaff's rules. The first experiment was DNA from different species. The first thing he did was isolated the DNA from different species such as E. coli, yeast, turtles, chickens, and humans. Oh, and a lot of more. It was a lot of different things that he was a really productive guy. Number 2, use paper chromatography to separate the DNA mixture. So, in paper chromatography, we have something to hold and is probably vertical, so you can hold up the paper. And you need a solvent. This paper, after you put the DNA here of different species, you can then put the paper in the solvent, and you would wait a little bit. The solvent will rise up the paper and start separating the nitrogen bases. Okay. After all of those nitrogen bases are separated, we can then use the UV spectrometry to measure the quantity of each base. The second experiment that he did was the same species but different parts. He isolated the DNA of the same species but took different parts of the DNA. So basically, let's say use humans for example. He took some cells from lungs, some cells from liver, some cells from sperm, and lots of more other things. And he basically compared the same exact species to itself, but like from a different body parts to see if they had different type of percentage of nitrogen bases. He then repeated the steps from the first experiment. The results and conclusions from this experiment can be graphed in this paper. There's actually a lot more that he did, but I wasn't able to put it up on one chart. But here is just the examples of some. So we have E. coli here, which is these three are types of bacteria. That's yeast, and then some animals, and of course humans. As you can see, here's the thymus, the liver, the sperm from humans, and different types of tissue that he used, and exactly which tissues he used. Here we have the adenine percentage, the thymine percentage, the guanine percentage, and the cytosine percentage. As you can see, the adenine and thymine percentages basically are the same, only like 1 or 2 up and down, but relatively if you round them, you get basically the same exact numbers. Same thing with guanine and cytosine. These two are matching. This basically confirmed what we said earlier, that adenine and thymine match up, and guanine and cytosine match up. Now, why does this matter? It led to the discovery of the structure of DNA and so much more. That's why it matters. We now know that DNA is a double helix. That purines, adenine, which are adenine and guanine, bind to pyrimids, which are thymine and cytosine, by hydrogen bonds. Adenine and thymine have 2 hydrogen bonds, and guanine and cytosine have 3 hydrogen bonds, making this one a little bit stronger. We also know that the width of the double helix is relatively consistent, because they have to be. That the 2 DNA strands must be complementary, because if you think about it, if they're pairing up, then one of them has to be paired up with the other, right? So one strand would be 5-3 to GCGGATT for an example, and then the other one would be its paired strands. So G pairs with C, A pairs with T, T pairs with A, C pairs with G. Now, because we know the structure of the DNA, we know its relative functions and everything that it can do. And if we know everything it can do, we can discover more things about it and more uses for it for our personal gain. And we have done this. In fact, we discovered the human genome. We use this for forensics, paternity tests, understanding genetic disorders, and so much more. So many of the things that we use DNA for example, for forensics, for example, we use that to catch criminals. We wouldn't be able to do that if we didn't know this. If we didn't know the structure of DNA, we would not be able to do half of the things that we do with it. And that was because of Chargaff and so many other scientists who helped in discovering the DNA structure. And that's what the beauty of science, in my opinion, is. So one scientist discovers a method, the other one does a couple experiments with those methods, and then they give their research to another scientist, and that scientist finds out more, discovers more things about it, and they pass that on over and over and over throughout the years. And then that's how we advance and we evolved. And it's a beautiful thing. And that is Chargaff's rules. Thank you. Have a wonderful day. And I will be linking my research paper, like the papers that I read and the videos I watched, and also the textbook that I used in order to conduct this research and this little demonstration. I hope I was a lot of help for anyone who is struggling with Chargaff's rules. Bye!